Okay, hello everyone. You can hear me all right. My name is Brandon, and I am the development manager here at Biocom Institute. Really excited that you all are able to join us for this session, collaborating with post-secondary and industry partners, the invaluable opportunities for authentic scientific experiences with Dr. Wendy Wooten and Dr. Gina Drake. Before we get going here, I will do a brief introduction and background on both of them. Feel free to send along questions, and we'll get to those as they sort of come through. Dr. Wendy Wooten is a science and CTE teacher at Reseda Charter High School. She earned her PhD in biochemistry and cell biology at UCLA, after which she completed an NIH postdoctoral fellowship in tropical medicine. She started her career in the Los Angeles Unified School District, teaching AP Biology, AP Chemistry, and AP Physics. She started her first robotics team at Chatworth High School and was a founding teacher at High Tech High, Los Angeles. Subsequently, she started a biomedical science pathway at Reseda Charter High School, where she currently teaches. And she believes strongly in authentic real-world research investigations for high school students to inspire them to pursue STEM careers in the future. And Dr. Gina Drake earned her BS in Marine Science and Biology from the University of Miami, an MS in Biology from San Francisco State University, and a PhD in Oceanography from Rutgers University. She previously was an NSF postdoctoral research fellow in biology at UCLA and remains at UCLA as an assistant project scientist and occasional lecturer in the ecology and evolutionary biology, Earth, planetary and space sciences, and atmospheric and ocean sciences departments, while also conducting research as a Zuckerman STEM postdoctoral fellowship at the University of Haifa in Israel. She studied and worked in coastal resource conservation, harmful algal blooms, seagrass, biogeochemistry, and the mechanisms and the evolution of coral calcification over the past 17 years. Thank you both for joining us. And just a quick reminder too, I'll remind at the end, but if you can rate the session inside the session, that would be great, please. Thank you so much, and feel free to take it away.
Okay, we're going to screen share here. I want to thank everyone who's attending for spending part of your afternoon. We're trying to encourage everyone to engage your students in authentic research because I think it's one of the most important aspects of the high school career for students that will be going into STEM. So our agenda for today, let me go into presentation mode. Our agenda for today, I'll just briefly talk about why we should be doing authentic scientific research. I'll go through some of the programs that we have introduced at Reseda Charter High School in the Los Angeles Unified School District. So you'll sort of get the perspective from the teacher's side, the high school teacher's side, and then we're going to have Dr. Gina Drake present on the research project that she does with our students, and so you'll get the perspective from a research scientist and somebody, an industry professional. So that's sort of the plan for today. Obviously, doing authentic scientific research is definitely going to improve the understanding of the scientific method and help develop critical thinking in our students. But the thing that I think that is most important is the fact that it's going to motivate the students because of the sense that they are contributing to the scientific community. They believe that what they're doing has purpose and meaning, and it serves as a motivation for them, more so than if they were just doing a classroom laboratory experiment. So I know that Amgen is doing a session on the Amgen Biotech Experience. This is the platform that all of our students begin their journey in doing scientific research. So they get to learn the basic lab skills, as you see listed, in a scenario that actually mimics authentic biotechnology. And so they see that it is an authentic situation. Then once they have these basic skills, because all of our ninth graders do the Amgen Biotech Experience, and then they also progress through the different levels of it in our biomedical science pathway. We have these other research opportunities embedded either in the curriculum of the pathway or as extracurricular research. And one of the first ones that they do that they build upon ABE is DNA barcoding. And this is a program that was developed by Coastal Marine Biolabs. This is an international science, citizen science initiative with IBOL, and the students actually publish a specimen page on the organism for which they're barcoding, as well as a sequence page. One of the characteristics of this program is the very high level of quality assurance and vetting. Normally, if students do sequencing, they just do one read, a forward read. Here, they do both the forward and the reverse read, and then they do bioinformatics to combine the two, and they have to look at the quality scores of the sequencing. This is very similar to another citizen science project which we've done and which is available, and that's the Wolbachia project. So these are both citizen science programs that are available and can be implemented in your classroom. In addition, and this is actually Coastal Marine Biolab's program. It's called Barcoding Life's Matrix. So if you're interested in, they will actually train you and provide the components and so on for this program, as does the Wolbachia program. Once we started doing DNA barcoding, we established a collaboration with CSUCI. I'm sure they don't want us to call it that. It's CSU Channel Islands. So once they found out that our students had been, well trained in DNA barcoding, they asked us if we would do a biodiversity monitoring on Santa Rosa Island, as the college was involved in the restoration of the island. So twice a year, our students go out, collect specimens, and then bring them back to the lab and do the DNA barcoding. Now, I want to point out that you can also incorporate a lot of these types of research projects within an interdisciplinary project. So another thing that we do is our 11th graders, the juniors, we have an interdisciplinary project where the U.S. history class looks at U.S. history as a series of immigrations and migrations within the United States. And to complement it, in our pathway course, we look at hominid immigration and migration over the world. So we do the out of Africa hypothesis, sometimes called the mitochondrial Eve, to learn about the different haplotypes. And then all of the students determine their own mitochondrial DNA haplotype. And by doing an ancestral family tree, many times our students are able to take the original hominids to their haplotype or all over the world, wherever it happens to be, and then all the way via looking at their family tree as much as they know how they got to Reseda Charter High School. So you can actually integrate these same types of studies as an interdisciplinary project with other courses. Having started some of these research projects and always reaching out to our local community colleges like we did with CSU Channel Islands, we also reached out to CSU Northridge or CSUN. And we connected with Dr. Cindy Malone. And they have a Build Co-Dare diversity program there for training students to do research. And she allowed our students to attend the summer boot camp. And in doing that, she was so impressed with their skills that many of our students were able to do research in her lab and other professors' lab. Also, they are associated with the UCLA CSUN stem cell and tissue culture program, and our students were then able to attend that course in the summer. So through this connection, our students have done summer research projects at CSUN looking at different transcription factors in cancer, looking at Vibrio cholera bacteria in copepods using qPCR and so on. So they were able to have all of these experiences through that connection with the local college. Another program that we've implemented is NeuroLab, and this is also with Coastal Marine Biolabs. And basically, this is a NSF-sponsored program where the students look at genetic diseases, neurological genetic diseases, and the genes that are involved in those genetic diseases. And it's part of the Human Brain Project that's a scientific citizen science initiative, and that's carried out with the Lawrence Berkeley Lab. So again, this is another citizen science project. It involves some recombinant DNA procedures, but also with using microscopic analysis and visualization tools. We also are in the process of introducing a synthetic biology and tissue engineering program. And this is a very current topic and cutting edge type of technology. And again, this is another outreach with a local university. This is with Dr. Morsa at USC. So see, we're being very fair. We're connecting with both USC and UCLA, so we're not biased. In any event, this program is to introduce the students to synthetic biology using the Notch signaling system, but also to look at how you can design genetic circuits to basically create different tissues. And associated with this is the iGEM competition, which we're hoping to join in the future. Another program that is been brought to us through Coastal Marine Biolabs is the Shellfish Aquaculture monitoring. And this is a project within LAUSD that's going to incorporate agriculture, public health, nutrition, and culinary arts. So it's a very interdisciplinary project, and it involves, in terms of our biomedical science pathway, it involves the monitoring of the water for coliform bacteria, as well as monitoring the shellfish tissue for various biotoxins, the paralytic shellfish poisons that we don't want to have in the food product, as well as domoic acid. So that's a program that's just beginning to start this upcoming year. So as you can see, the programs that we have that really give students the opportunity to conduct research and to have the experiences where they're mentored by research scientists, industry professionals, all center on the universities and companies and organizations that are within our area. And they all all of these programs were put together just by reaching out and connecting with them and bringing these opportunities to our schools. At this point, are there any questions before I introduce and have Dr. Drake talk about an additional program that she brought to our high school? Are there any questions about any of these particular programs that are, and similar ones are available to, I'm sure, every high school?
I don't have any questions yet, but I will monitor and let you know.
Okay, so like I said, if you contact Coastal Marine Biolabs, they've got the DNA barcoding, the barcoding lights matrix, as well as NeuroLab. That's a very strong NGSS program with a lot of modeling and research opportunities, and also this aquaculture program. You can contact Coastal Marine Biolabs, and then different universities or colleges that are near you. That's where you can get a lot of research experiences and opportunities for schools. And now that we have one other program that's been going on a few years, and this program was brought to us by Dr. Gina Drake, who is from UCLA, and she actually contacted our school and to see if we would be interested in it. So again, this is where the universities are beginning to reach out and connect with high schools, because they're beginning to see the value of well-trained high school graduates who then can come and continue. So at this point, I'll turn it over to Dr. Gina Drake from UCLA to talk about our proteomics program.
Actually, Dr. Wooten, quickly, I'm sorry, there's a question from one of the attendees. Can you elaborate on the funding mechanisms for these programs, please?
Okay, actually, most of these programs in our situation, like, okay, Amgen, the ABE Amgen is free. They provide all of the equipment, all of the supplies, they train teachers, and so on. So that's free. The other programs, we're a CTE pathway, and everything is covered in our Perkins grant. Every year we apply for Perkins grant, and all of this is covered through our Perkins grant. When we have students go to the universities and work with them, the researchers at the universities are the ones that are picking up the costs on it, because basically they're bringing students in to do lab work. I know that a few years back, we actually had a student that went to UCLA, actually two students that worked in a lab at UCLA. The contact there, she worked in the human microbiome, and our two students did a project on microbiome and acne. Who else would study acne but high school students? Anyway, they used next-gen sequencing, which is really expensive, but basically because they were working in the university, it was part of the professor's grant money. So, you know, other than just having our annual Perkins grant, you know, that a lot of the research is paid for by the organization, the college or whatever that is sponsoring the program. Okay, any other questions?
I do have another couple here. When you reach out to a potential partner, do you look them up and email them or call them? What's your outreach process, please?
Okay, so first of all, you have to realize that you're going to hear no many times, but the thing is you use every opportunity you can. Basically, you want to make these people your best friends if they have something that's going to be valuable to you. So, for instance, if like we've done some RNAi lab projects, and so I would Google like UCLA and see who was working with C. elegans doing RNAi, and so then I would contact them and say, you know, would it be possible for you to supply us with the, you know, the worms and the feeding bacteria and so on. And they've all been really good about doing that. I mean, attending, one of the things is attending symposia like these or workshops and connecting with people, and then you know, just asking. I know Biocom sponsored a workshop with Illumina, and I connected with some of the people from Illumina, and they became judges for our capstone project. I requested having DNA sequencing done by them, and even potential donation of equipment. So basically, just every possible manner of you know, connecting with post-secondary institutions, vendors, research organizations, and so on, and then just asking them for the things you need. Other questions?
Great, thank you. I'll hold till the end and send it along to Dr. Drake here, and we'll follow up if there's some time at the end.
Oh,
thank you.
Okay, cool. So this is just the about the aquaculture. Oh.
Thank you, Dr. Wooten. So hopefully you've been able to tell from the slides that Dr. Wooten went through that she has done a phenomenal job putting herself out there to get all of these opportunities for these students. Reseda High School is a public high school in the pretty much smack dab in the middle of the San Fernando Valley. And it's not a private school or anything. These are just kids from the Valley, and she's gotten them some amazing opportunities. So big shout out to you, Wendy, for that. So as Wendy mentioned, I'm at UCLA and I study proteins. Specifically, I try to sequence proteins from organisms that make some sort of biological hard part. And in 2018, I was looking for an outreach program to kind of establish as part of my National Science Foundation postdoctoral fellowship in biology. The National Science Foundation is currently really trying to ramp up their outreach opportunities of people who are funded by the NSF. They've done this for a long time, but they're really pushing it strongly now. So the outreach isn't just lip service, but there's actual service being done to the community, broadly defined. So UCLA has affiliations with several high schools close to the campus, and several of them are private schools. And I don't live in Westwood. I live in the middle of the San Fernando Valley. So I thought, well, where's my community here? How can I take what I do at UCLA to students where I live? I found Reseda High School, and I emailed the principal and said, here's a program that I'm proposing. I'd like to teach high school students how to handle protein sequence data, very complex data, have them work through some of the data with me. I'm then able to use the data and they're able to learn a skill. And so the principal of Reseda High School got me in contact with Dr. Wooten, in addition to her being very proactive and reaching out to people to find these opportunities. She's also great at saying yes. She said yes to me. And so I've been working with her students for the past 3 years. We're now on our 3rd cohort of students, where the original cohort, who are graduating seniors this year, now are able to act as mentors to our first-year students who are our mentees. So the students analyze protein sequence data so I prepare the samples, I submit the samples for sequencing. My NSF fellowship paid for the sequencing and all of my supplies. And then I bring the data to the students. And we would meet weekly, 1 to 2 hours before the pandemic shutdown. We would meet in Dr. Wooten's classroom at Reseda High School. The students would get into groups and they would analyze the data through a couple of different programs that I taught them. The first is listed here, Mascot. This is a proprietary program. The Proteomics facility at UCLA pays for a license, and with my affiliation with UCLA, I am able to have access to that license. The NCBI BLAST tool is free, and it is becoming more widely used in university, like undergrad class settings. So I consider that to be a transferable skill that those students are learning. And then some of the data management is done in Excel, which I think is also an important skill to have, to be able to use Excel extensively. Other than me driving to Reseda High School, there was basically no cost to this program. We use resources in Dr. Wooten's classroom. Through a grant, she was able to secure several laptops previously that we're able to use for the project. And then we just hook up to the Ethernet with Ethernet cables, and we make a maze of Ethernet cables that I have to hop around. And then my access to Mascot, which made for some fun times because I don't have signal in Dr. Wooten's classroom. So I've had to send students outside with my phone to answer my phone with the multi-factor authentication to use the UCLA access. When everything shut down with the COVID-19 pandemic, we were able to continue over Zoom. And I asked the students, this is a hard time. Do you just want to give this up? And all of them chose to continue with it, which says something about the quality of students that Dr. Wooten teaches. Next slide, please. As part of, was that last year? Time doesn't exist anymore. Last year, as part of the proteomics program, Dr. Wooten was invited to have some students submit grant proposals to the Archer STEM research program. And so the Archer School is a private school in Los Angeles that uses part of their funds to let students from other schools apply for these research grants. We had 2 groups of female students, 3 students each. Each group secured a grant worth over $1,300 to do their own research that they proposed. One group worked on coral skeletal proteins, going down a core. Another group worked on fish otoliths or ear bones in fish, looking at both modern and fossil otoliths, and both groups sequenced, they went through data, proteins extracted from those 2 biominerals. As part of that funding, they were able to also use public transportation to visit UCLA. They've met with the person who runs the proteomics facility, shown in the black shirt in the top picture, Dr. Julian Whitledge, who's been incredibly supportive of interacting with the students as well. The students were able to visit his lab. They were able to visit the labs where I do my sample preparation and protein extraction, and they were also able to interact with graduate students during that visit. On other visits, they were able to visit the UCLA Meteorite Gallery, the Scanning Electron Microscope Lab, and the Ion Microprobe Lab. The Ion Microprobe is a fantastic instrument. It's a mass spectrometer. There are not very many of them in the world, so this was a unique opportunity for them to meet the person who basically built this instrument several decades ago. And again, interact with graduate students and postdoctoral researchers. Normally, as part of the Archer STEM research program, the students would go to the Archer School in May and give a presentation of their research. Last year, obviously, that didn't happen in May. And so I said to the students, well, you could present, you could do something over Zoom and record it, and then we could distribute that video to your family and your teachers and your principal. And they all were super jazzed about it. And they, each group made about a 10 to 15 minute long video describing the motivations behind their research, what they found, and what the implications are. And I uploaded those to YouTube as an unlisted video that they then had access to the links for that they could share. And their principal was super proud of them. And then lastly, some of that funding went to take the students to do some coastal lagoon monitoring with another group that I'm affiliated with at UCLA, Dr. Dave Jacobs' group. They visited 2 lagoons in Malibu and helped to survey like lagoon water depth, height of the sand berm, and they also collected some samples for environmental DNA or eDNA that is part of a PhD student's project. So the students were also able to interact with more PhD students through that and also have a fun day outside along the beautiful California coast. Next slide. So as I mentioned, I offered the students during the shutdown last year, we can all just take a break, and they wanted to continue. We continue over Zoom. We meet weekly. We actually met today at 3 o'clock over Zoom for an hour. Because Mascot is proprietary, I can't give them access over their personal laptops. So what we do is we pivoted to characterizing proteins based on their sequence. And so they're learning to use some free online and downloadable softwares, including a ProteParam, which gives you some quantifies basic characteristics of proteins. They use Signal P, which is a website that predicts the signal peptide or the traditional mechanism for telling a cell to export a protein. They use TMHMM, which predicts transmembrane regions in proteins. And the idea is that they'll put all of this information together in a spreadsheet. They each have 10 proteins that I assigned them. They'll carefully characterize all 10 of these proteins, and then based on what they find in their spreadsheet, what different interesting patterns they find, they will choose one of their proteins. So I phrase it to them as, you're a researcher, you have limited funds, characterize your 10 proteins. You only have funds for one protein that you are going to pretend to crystallize and then model and analyze. They'll choose their one protein each. They'll use a free program called iTassr to model that protein. And then they're also currently now working with a free for educational use program called Primal, where they'll put the modeled protein into Primal, and it allows you to have like a 3-dimensional view where you can rotate the protein and you can color different parts of the protein. You can color it based on amino acid or something like that, so that they can visually see their protein of interest. And I should say, we have, some colleagues and myself have written this program into a proposal that we currently have under reconsideration by the National Science Foundation. And we've got our first batch of feedback back from NSF. They had their criticisms of, of things they wanted us to clarify of the science, but we had 5 reviewers. All 5 reviewers were so jazzed about this project. They thought it was a really neat thing, and hopefully this NSF proposal gets funded, because the next step then would be to start to analyze maybe students' grades in certain classes that proteomics is important to see if the program is helping their grades improve at all, or helping them understand different concepts more than students who are not participating in the program. And so with that, I'm happy to take any questions.
I have a question here that goes back to the DNA barcoding. And it's, do you send the samples out for sequencing, and how do you get started? Are there protocols at the Life IBOL site, please?
Okay, so for DNA barcoding, basically to have your own sequencing facility at the school probably is not going to happen, but that's where reaching out to places like Illumina that, I said I connected with over at a symposium, being able to ask them, can you sequence these for us? We've done that with, hang on, there's a sequencing, I can't remember. We basically asked a sequencing service for free sequencing, and they did it. We've also asked SnapGene for use of their. It's a company that has, it's a bioinformatics platform, and they've let us use it for free. So very often you can request free services and they will grant them to you. That cost, even if you do send out for sequencing, we get forward and reverse sequences for about $7 per sequence. But one of the things that's important about the DNA barcoding through Coastal Marine Biolabs, there's a lot higher quality control. In other words, instead of just showing that you have an amplicon band on your gel, you have to be able, it has to have a certain brightness where you have to quantify it, the amount of DNA to make sure that it's a good sample and you're not going to waste the sequence services. So one of the advantages is the students actually get to see that in actual research, you do have a higher bar set than a lot of labs that you can purchase for classroom use, where you send them out for sequencing, you only do a forward read, and they don't really care whether you have a good sample or not, and then you just get the ones that work back, and the other ones they just say no sequence. So we've been very fortunate in getting a lot of this stuff done for free just by asking. So for example, with SnapGene, I just emailed them and I said, we're having this project, we need to do such and such. Would you give us a month of access to your bioinformatics platform for our students to use? And they said, yeah, anytime you need it, let us know. So there's a lot of industry support and even piggybacking on all of the things that Gina talked about with the proteomics. These kids are going to go off to college and have so many skills, technical skills. But the other thing is, she taught them how to write and secure grant funding, which, very few high school students, get their own grant. So they're heading out with technical skills and professional skills, because of this collaboration between research scientists and industry professionals with the high school. So yeah, it's just a matter, people want to support this. So all you have to do is ask.
And to add to that, like I mentioned, NSF and other funders too are very strongly pushing concrete outreach activities as part, the funding is contingent on having these outreach activities. And a lot of people don't necessarily have the outreach idea developed yet. And so if you have an idea and you have someone that you can reach out to, the people who are writing the proposals would be super excited to have someone with an idea already in mind that they can contribute to the development, rather than trying to come up with their own thing from scratch and then having to do what I did of find the person to do it with.
Great, thank you both. And another question here. Were there any challenges or intentional scaffolding involved in getting students to absorb these types of science content? Basically, how was the students' learning curve towards the proteomics work, please?
Okay, yeah. So that kind of occurred with how everything developed time-wise. So we started with Amgen ABE 10, 15 years ago when they first started. So that's where they started learning their basic skills. Well, at that point, you know, by reaching out and saying, these students, they know how to create recombinant plasmids. They know how to transform bacteria. They know how to express genes in a recombinant plasmid and purify the protein product. You can start talking to, and especially community colleges now have a lot of programs, and they really want to interact with the high schools. So you can start talking to post-secondary professors and start creating these programs. We did that with the DNA barcoding with CSUCI out on Santa Rosa Island. We did that with the interaction with Coastal Marine Biolabs with their other programs, the NeuroLab and so on. We did it with CSUN. We did it, you know, like both USC and UCLA with the proteomics and the synthetic biology. They came to us. I know the synthetic biology program that we're developing now from USC, that occurred because the professor heard about the other things that we had done. So it's a thing where you start with something basic, you know, like the Amgen ABE, and then you just build on it. And as opportunities arise, you know, you just go from there. So, you know, like I remember our kids presented some of their work from DNA barcoding at NASA's Regional Science Fair, and students from other schools said, oh my gosh, we just learned about, you know, doing DNA sequencing and using it to develop phylogenetic trees, and you guys are actually doing it. So it's just a thing of building, you know, based on the skills that you're bringing in for the students, and them, you know, just moving to the next level and the next level. And, you know, proteomics in essence is just, you know, a slight divergence from doing DNA sequencing and so on. So there's those connections that make it have, you know, a way to scaffold into proteomics. And, you know, obviously, you know, Gina introduced these different bioinformatic programs one by one, but it's a thing, you know, where it just builds upon the knowledge that they're getting as they learn.
Thank you so much. And I think we have time for one more here. We're getting close to the 5:15 ending. I noticed that PYMOL, I'm not sure, Primal, has a free version and a classroom with instructor students license. Do you still recommend the free version, or is a license going to give you something more worthwhile?
Sure. Yes, you did pronounce it correctly, Primal. So the free version, you just on their website have to put in your affiliation and what you're going to use it for, and then I guess they check your affiliation and they'll send you like a key code. So anyone can download Primal for free, but the first time you use it, you have to put in your user key code. The free version has a time limit to it. So they gave me about a 6-month time limit, and then they emailed and said, you know, if you want to extend that, we'll have to have a discussion. So they may not let you have the free version forever. But as far as I can tell, all of the functionalities, the basic functionalities of Primal that you would use at the high school or probably even an undergraduate level are the same between the educational and the paid license version.
Great. Thank you both. We are right at 5:15. Thank you so much, Dr. Drake and Dr. Wooten. That was really fantastic. We really appreciate you taking the time. And thank you to everyone who attended. Really appreciate it. If you can all rate this session, it's here. Before you leave, that would be great, please. And we hope you can join us tomorrow. It starts at 3 o'clock. Thank you all again, and especially thank you, Dr. Drake and Dr. Wooten. We really appreciate it.